Okay, can you guys hear me?
Yeah.
No, you will watch.
Yeah? Okay,
Yes.
okay, perfect. Okay, can you guys see this PowerPoint?
Yes.
Perfect. All right, any questions about anything we've covered so far? All right, I'll take that as a no. So we left off talking about the carbs, and we talked about that carbohydrates are made up of lots of subunits of what we call monosaccharides, and then those monosaccharides can be linked together to make disaccharides. Do you guys know what the reaction to link monomers to make polymers is called? So you make sure you know these reactions for the exam. Dehydration to build molecules, hydrolysis to break them down. And so that's what we talked about here in the reaction. Okay, so anyway. So starch is long chains of glucose, and we talked about how we number these. So so they're numbered from the oxygen that's within the ring, and we go clockwise. So this is number 1, number 2, carbon, 3, 4, 5, and 6. And because it's 6 carbons, then it's a hexo sugar. And this particular sugar is glucose. I can tell by the way that it's built. Other ones might be have a pentagon ring with 2 carbon attachment groups here, like this carboxyl groups. Anyway, so this is 1, 2, 3, 4, 5 here, and 6. So this is a 1,4 linkage. And we call it an alpha linkage because the orientation of this is alpha glucose. So remember we talked about double bonds? If we have a double bond and 2 oranges on each side of that, we can't spin the oranges because there's 2 sticks there and they hit each other. So think about it this way. If you had an orange, so this is my orange, and I shove 2 sticks in it at an angle, like these ring structures have, would I be able to spin this orange?
No.
No. And so because it's a ring structure, it acts just like a double bond. And remember, these double bonds cause geometric isomers. Which are different. So these hydroxyls give it away, these hydroxyl functional groups, they're on the same side. And so if they're on the same side, what do we call that? It's either cis or trans.
Cis.
Cis is the same side. Good. Now, in beta glucose, this hydroxyl is up here instead of down here. And so then they're across from each other, not on the same side. This one's up and that one's down. And so what would we call that? Alpha, that beta glucose.
Trans.
Good. So that's why it's important that you guys understand these geometric isomers in cis and trans, because these linkages are going to make completely different molecules. One you can digest, the other ones you can't. And that's what we're going to talk about now. So that makes the difference between whether these polysaccharides are for energy storage or for structural support. It has to do with either the alpha or the beta linkages. Okay. So to carry on with starch, it's glucose. It's a polysaccharide. This is only found in plants. Right? We don't store, animals don't store their energy as starch. How do we store our energy?
As glycogen?
Well, yeah, glycogen is right. So somebody's read ahead. That's good. But generally, our long-term storage is fat. And there's a reason for that. Fat has a lot more energy per gram than starch does. So if you stored your energy as starch as an animal, you would weigh about twice as much, which means it would be twice as hard for you to run away from an animal that stored its energy as fat. Usually you would get caught and then you would be killed, and that survival of the fittest would go away. So all animals have developed fat as an energy storage. Why wouldn't plants use fats? Frankly, they don't care because they don't have anywhere to run. So they don't care what they weigh. There's no advantage or disadvantage to that. So in starch, in plants, right, I might ask you on the test, where's starch found? Is it found in, is starch found in shrimp? No. Is starch found in lions or tigers or bears? The answer is no. Remember, it's only in plants. That's their energy storage. This is made by alpha 1,4 linkages. I don't care that you know the structure of this. I'm just trying to explain it to you. But I do care that you know that this is an alpha 1,4. So if I ask you on the test, what's the linkage in starch, you need to be able to tell me that it's an alpha 1,4 linkage. Okay? Now, there's two different kinds of starch. One is called amylose. Remember that OSE tells you it's a sugar. This is unbranched. So this is amylose right here. It's just a linear chain. It takes on this sort of helical structure because of the constraints of the bonds, just the way it's built. You can sort of imagine if your Lego pieces had a certain shape, like they bent, then when you built your Lego tower, it would start to curve. And it's because of that. It's just the structure of these molecules that cause it to bend that way. So amylose is not branched, it's linear, and it forms a helix, which is shown here. And then the other form of this starch is we have amylopectin. This is the same sort of pectin that they use to make jellies and jams and stuff like that. So this is branched form of starch. It's a little more complicated, and you can see here that we have the typical helix for, say, amylose, but you can also have these branch patterns that come off of it. They're still alpha 1,4 because it's still starch. So on the test, I might ask you, is amylose straight or branched? In other words, is it unbranched or branched? And you should be able to tell me that it's unbranched. And then amylopectin, I might ask you the same question. Is it branched or unbranched? And you need to tell me that it's branched. I might ask you, what's the linkage between amylose glucoses? And you should be able to tell me they're alpha 1,4 linkages. I might ask you, what's the linkages of amylopectin? And you should be able to tell me that it's alpha 1,4 linkages. This is an electron micrograph picture. So electron microscope took this image. This is a chloroplast, and you'll learn more about these when we get to chapter 6. And then you'll learn again about it in chapter 10 when we do photosynthesis. But just for now, chloroplast is the site of photosynthesis. And in photosynthesis, what plants are doing is taking carbon dioxide and making glucose. these monomers, and then they take the glucoses and they link them together in long chains, and they make starch. And that starch is what they use to store energy. It's very similar to what we talked about in solar panels. So if you had solar panels on your roof, you would be making electricity, but only when the sun's out. When the sun's not out, you can't make electricity. So you have to have a way to store that energy to make it through the night. So for us and our solar panels, we would need batteries. For plants, they use starch to store the energy, and then they can do what you do, which is break down the starch and release it as energy. So they basically eat their own food at night that they've made during the day from carbon dioxide. And that's sort of how and why photosynthesis occurs. All right, you guys have any questions? So when you're making, if you're gonna store the energy from photosynthesis, and photosynthesis occurs in chloroplast, you want to store it near where you make it. So again, if I had solar panels on my roof, I wouldn't want to store my electricity 6 miles down the road. I'd store it in my garage because there's less wires I have to run, there's less distance, I won't lose any energy by sending it that far away. So plants store their starch in the same site that they make it through photosynthesis in the chloroplast. Any questions? All right, so we'll go on to the next. So just a reminder, starch is made in plants only, right? They store this in chloroplast. This is just a general term for these plaster regions found in plants. Plants can store extra glucose as starch and use it as energy, like we just talked about. Like during night, they do respiration just like we do. And I know that this is a little weird because, you know, whenever I was in early biology, I kind of thought that plants, you know, that humans breathe oxygen and they gave off carbon dioxide, and plants breathed carbon dioxide and gave off oxygen. And this is that timeless wonder that, you know, we're giving plants CO2 and they take the CO2 and make oxygen and give it back to us. But really, plants do both. They can do both of these. They do this at night when there's no energy available, and this during the day when they can use photosynthesis to generate energy. So this process is called taking in oxygen and giving off carbon dioxide is called respiration. And this process, taking in carbon dioxide and giving off oxygen, is called photosynthesis. And we're going to learn about respiration in chapter 9, and we're going to learn about photosynthesis in chapter 10. So for right now, for the test, I might ask you, where do plants store their starch? And you would tell me chloroplast. Plants can store glucose as starch, and you know they could use it as an energy source. They can also build other molecules from it. Remember, we talked about plants have to make proteins, so that means they have to make amino acids, and they can't eat them. I mean, some plants do, like Venus flytrap, but they're the exception. You don't see like pine trees going around eating other animals or organisms. So because of that, they have to take the carbon dioxide and combine that with amino nitrogen from the soil to make amino acids. Like that. So they have to turn this into this. And that's how they use it as a carbon source or a carbon skeleton, which I discussed earlier. Animals that feed on plants can also access this starch for their own metabolism by breaking down the glucose. And we'll talk about this in respiration and how that occurs. Any questions? Okay. So glycogen, and sorry about that. Another one. Okay, so glycogen is a glucose source that's found in animals. So remember, starch is plants. And we talked about the kind of starch is amylose, which is straight chained, and amylopectin, which is branched. So make sure you guys know that. In animals, we use glycogen. But remember, starch is heavy. It's much heavier than fat. In fact, if you looked at your nutritional label, you would see that for every gram of carb, it's about 4 calories. And for every gram of fat, it's about 7 calories. So you get almost nearly twice the amount of energy per gram from fats that you do from carbs. But the problem with fats is that in our bodies, the fats are in our adipose cells, our fat cells. So let's just imagine that you're out in the woods and you come across a bear, right? And so this bear decides it wants to eat you. So you could play dead. Sometimes that works, maybe not. Maybe the bear will eat you. Most people would run. And so think about if you stored all your energy as fat, your body would have to release the fat from your fat cells. It would have to travel through your blood. It would have to get converted into our energy currency, which is ATP. And we'll talk about this in respiration too. And then you would have to have that ATP go to your muscle cells, because that's going to allow you to run away from the bear. That takes a long time. And by the time all that happened, you'd already been eaten. So we have to have an instant ready supply of energy in case something crazy happens, like we need to run away from a bear. And that instant energy is glycogen. We don't want to store a lot of this because it's heavy, like it's still like a carb. But we don't want to store a lot of it because it's heavy. So we store about a 24-hour supply. So it's an emergency supply of glycogen. And usually that's stored where we need it the most, in the liver and in your muscles. So that's what I've written here. And again, this is found only in animals. It's highly branched alpha 1,4s, like we talked about. So this is alpha 1,4 bonds. This is alpha 1,4 bonds. In glycogen, we have alpha 1,4 bonds, but we also have alpha 1,6 bonds. Remember, that's just between the number 1 and the number 6 carbon. I'm not going to draw it out. You guys can look at the other slides to figure out where the bonds are, but that's the difference. And you can see here, this is an alpha 1,6 linkage. Here's an alpha 1,4 linkage. So they can have both, but the main point of all of this is all of these, they have alpha bonds in common. And alpha bonds, animals can break down readily, like humans. Okay, so on the test, I might ask you, what's the bond in amylose, like I already told you, and you tell me alpha 1,4. I might ask you, what's the bond in glycogen, and you should be able to tell me it's alpha 1,4 and alpha 1,6. I might ask you, well, is glycogen found in plants? And what would you tell me? No, the plants use starch. Well, is starch found in animals? And the answer is no. Animals use glycogen. So make sure that you're able to answer those sort of questions on the exam. Now, this kind of brings up an interesting point. I'm just going to sort of let you know why this is important. You guys ever gone to the gym and looked at like the elliptical or the treadmill or something like that? And then it has like a cardio zone and a fat burning zone. So which one of these do you have to work harder at?
Fat burning?
You think so, but next time you go to the gym, take a look. This one is low level. So you want to keep your heart rate around, well, it really depends on what your sex is and your age and stuff, but this is generally relatively low, like 110 to 120 beats per minute, where this might be closer to 150. So you would think that if you ran your ass off on the treadmill, that you would burn fat, but you would be wrong. And the reason is because what's your instant energy source? The one, if you're running really fast in a treadmill, your body thinks that you're running away from that bear. So what are you going to use as an immediate energy source?
The glycogen?
Right. So you're going to burn glycogen if you run 150 beats per minute or higher on the treadmill. Whereas if you're going slower, your body doesn't think you're being chased from a bear, and it thinks it has time to release the fat to burn it for energy. So lower level activity actually burns more fat than higher level energy. And the reason is because of biology and because of glycogen. Now, if you burned up all your glycogen, then you would have no other option but to burn fat. But you would have to fast for like 24 hours before you could do this. Or you could, you know, put your body in a state of ketosis or something like that, so you don't eat any forms of sugars or glycogen at all. But that's the difference between the cardio and fat burning on your treadmill or elliptical or bicycle. All right, any questions about that? All right, so those are the energy sources. Now we're going to talk about the structural sources. So if you look on your nutritional label under carbs, you'll see that there's a section called fiber. And fiber is the exact same thing as cellulose. So if you wanted to get some fiber, you could go eat some tree bark or whatever. It's basically the same thing. It's the undigestible part of plants. And so cellulose is actually glucose, but it's the beta form of glucose rather than the alpha form. Remember we talked about alpha earlier, and these hydroxyl groups are on the same side, so they're both down. So this is cis, and here in the beta, they're across. So this one's down and this one's up. This is the plane that it's in. And so this one is actually trans. So cellulose is glucose. It's the same molecule, except this one is an isomer of this one. Does that make sense? So we have beta glucose and alpha glucose. They're both linear, so they're unbranched. But when we link beta glucoses together, they sort of look flat. Remember when we linked alpha glucoses together, what was the shape? Now let's go back. So these are alpha glucoses linked together. What's the shape here?
A helix?
Right. So with beta glucoses, we get this flat, this linear structure. And it's because they go up and down, whereas the other ones go up and then they go up again. And by doing this, you're forced into this helix structure, like so. So alpha glucose is more of a sort of a helix. Beta glucose is flat. Now you might be like, what's the difference, or why is that important? And I'm going to show you in a second. But what I want you to remember is that cellulose is called fiber, right? It's made out of glucose, so it's an isomer of glucose. The only difference is that it has beta 1,4 linkages instead of alpha, and that it's linear, and it makes this flat shape. That flat shape is going to cause it to, remember the most important thing in biology is shape. Nothing is more important than shape of molecules. If it has a different shape, it's going to have a different biological function. And the same thing is true with this. So I'm going to skip ahead and I'll come back to this. So here's the shape that cellulose makes. It's these flat shapes because of the beta 1,4s. Remember, they go up and down, up and then down, up and down, which causes them to be linear. And remember, we have oxygen and hydrogen on these, and you guys did this in the chemistry lab. What's this functional group called?
Hydroxyl.
Yes, you guys make sure you know this for the exam. I will draw, I promise you, I'm gonna draw this on the exam. I'm gonna ask you what that is. So that's a hydroxyl. Do these share equally or unequally?
Unequally.
Yes, remember the electrons stay around oxygen a lot more than hydrogen. That makes hydrogen have a partial positive charge and oxygen have a partial negative charge. And anytime we have oxygen and hydrogen, or nitrogen and hydrogen, you guys should instantly think these things are going to form hydrogen bonds. As a G, not a D. Hydrogen bonds. So this hydroxyl is going to link to this hydroxyl in these long chains. So think about if you had a rope and you're it's basically getting braided from these hydrogen bonds. So every time you add these hydrogen bonds in here, it makes it a little stronger. And if you're braiding them together, then you can make it really strong. So I saw this Mythbusters episode where they actually climbed out of a third floor of a jail by using toilet paper that they had braided together several hundred times. And because of this flat shape, it can make all of these hydrogen bonds and these, and I don't know if you guys, since you don't have access to a microscope, usually I would have the students, when I'm talking about this, look at a piece of paper under a microscope, but this is what it looks like. And this is what gives plants, the cell walls of plants, their super strength. And if you don't believe that a plant cell wall is super strong, just go run as fast as you can into a tree and see which one goes down, you or the tree. So these bonds, these multiple hydrogen bonds, give these the cellulose molecule incredible strength. And it also makes it hard for you to digest it because of the shape of these bonds and the way that these bonds are interconnected with one another. All right, any questions about cellulose? So on the test, remember cellulose is made up of beta 1,4 bonds. These are indigestible by most organisms. So, you know, we're not talking about cows and stuff like that, but for the most part, most animals can't digest this because of the structure. And that these beta 1,4 bonds make lots of hydrogen bonds that give it its strength. All right. So chitin is the next one. This is also structural, but it's used by insects. So insects like arthropods, those are spiders, crustaceans, insects. They use chitin. So it's similar to cellulose. It's a glucose. It also has beta 1,4 bonds. The difference is that there's an amide group rather than the hydroxyl group on the second carbon, which is shown here. So remember 1, 2, 3, 4, 5, 6 carbon. So it's on the number 2 carbon. And you can see it here. And then this also alternates. So this is up and this is down. And so that makes the structural support for insects like the hard shell of lobsters and crabs and things like that. So just to summarize, so we have the carbs, right? Which we also call starch, same thing. And we have simple carbs, which are the monosaccharides. And the disaccharides. And then we have the complex ones. And in those complex carbs, we have the starch, and then we have glycogen, and we have, and in the starch we have amylose and amylopectin, and then we have cellulose, and we have chitin. So on the test, I might ask you, for starch, is amylose straight, chained, or branched?
Straight.
Right. And I might ask you, is amylose an alpha 1,4 or a beta 1,4, an alpha 1,6, or all of the above?
It's a 1,4.
Right, alpha 1,4. Good. And then amylopectin, is that straight or branched?
Branched.
And is that an alpha 1,4, alpha 1,6, beta 1,4, all of the above, combination of any?
Is that just 1,4 or is it?
It's an
1,4
alpha.
and 1?
It's an alpha 1,4, but it's branched. Same
Yeah.
linkage, branched. Perfect. So you get those right on the test. Glycogen. Is this glycogen? Is it straight or branched?
Is it branched?
Yes. Is it alpha 1,4, alpha 1,6, or beta 1,4, or a combination of these?
Alpha 1,6.
And
116.
yeah, alpha 1,4 and 1,6. So you need to add both of those in there. Perfect. Cellulose. Is this straight or branched?
Is it branched?
No. It's linear chains. So just like we showed here.
Okay.
All right.
See you next time. All right, bro.
All right,
Study
son.
too hard.
All right, son.
Good luck.
All right, son. Thank you.
So this is straight. Cellulose is straight chain. And then what's the bonds in there? Alpha 1,4, alpha 1,6, beta 1,4. Good. So these are beta. Chitin, same thing, right? It's beta 1,4, and it's also straight chains. So I might say, well, animals can only digest alpha linkages. So of all of these, what can animals digest? If we can only digest alpha linkages.
Starch.
Starch and
Glycogen.
glycogen, good. It can't digest cellulose or chitin because those are beta linkages. I might say, well, we found an organism on Mars and it can only digest beta linkages of these, which could it digest?
Cellulose and chitin.
Could it digest starch?
Oh, I see. What, you need me to tie it?
No.
Yeah, that's right, because starch is alpha and this thing can only digest beta. Good.
Okay.
So this is the kind of things that you'll hear on the exam, or you'll see on the exam. Okay, so that covers the carbs. Remember, we're just sort of covering the things on your nutritional label. So we're going to go over the carbs and the fats and then the proteins. And then we're going to look at the DNA and the RNA, the nucleic acids. So we finished carbs. Now we're on to fats. So fats we call lipids, right? In general, there's different kinds of fats. We have regular fats. This is called triglycerides. The real technical term is triacylglycerol, but I don't care if you know that. Then we have phospholipids. These are special fats that have a phosphate functional group on it. And we talked about phosphate functional groups in chapter 4. And then the last group of lipids are the steroid hormones. So what these all have in common, and most of you guys got this right on the chemistry one, is that they're nonpolar because they're made up of carbons and hydrogens. We call these hydrocarbons. And that means that they're not dissolvable in water. All right. So nonpolar covalent bonds. And they're hydrophobic. Any questions? Okay, so let's take these each individually. We're going to start with the triglycerides or the fats. This is what you guys did on the chemistry lab. So a fat is a macromolecule that's composed of glycerol, a glycerol head, call this the head, and they're linked to fatty acid, usually 3 fatty acid. We call those tails. So fatty acids are long chains of carbons and hydrogens, right? Like, you know, longer than what we talked about, like octane and methane. So methane would be 1, ethane is 2, propane and butane. And then we talked about if we had 8, that would be octane. So, you know, those are like combustible gases. Fatty acids are usually, you know, 14 to 23 carbons long, but these can vary. And the length of these makes it different. So if you ever looked at like canola oil and sunflower oil and coconut oil and these different kinds of, you know, rapeseed oil and stuff like that, olive oil, they all are made up of different length fatty acid chains. And that's what makes those oils different. So all of these triglycerides have in common that they have glycerol heads and 3 fatty acid tails, which are linked to this glycerol. And then you can go back and look at your chemistry lab and see that you guys did this. So if we want to link these together, we have to take out water. What's that reaction called?
Dehydration synthesis.
Yeah, make sure you guys know that for the test. Anytime we build molecules, we're dehydrating them and we want to break them apart, we have to add water. That's hydrolysis. So fatty acids, like we said, they're carboxyl. So it has this carboxyl group right here. I might put this on the test and ask you to identify this functional group. Make sure you guys know what that is. It's the same one you identified on your lab. Attached to a long carbon skeleton. So 16, 18, like I said, it can range anywhere between 14 and 23. And sometimes even longer. I don't care that you know that this is an ester linkage, but this particular linkage right here would be considered ester. Don't worry about that. I'm not going to ask you this on the test. So, but you should know that triglycerides are made up of, that word tri means it's made up of 3 fatty acids. And that glyceride comes from the fact that it has this glycerol head, which is basically methane, ethane, propane. So it's actually propanol, but because it has 3 hydroxyl groups on it, it's called glycerol. And that OL tells you it's an alcohol, and that tells you it has these hydroxyl groups on it. All right, so this is the ester linkage, which is more clear. And then here's the hydrocarbon chains of the fatty acids, which are here. So tri, 3, acyl has to do with this acetylide region, and then the glycerol is this molecule here that they're linked to. So triglycerides or technically triacylglycerol is a fat that's made of 3 fatty acids and 1 glycerol linked together by ester linkages, which I don't really care if you know that or not, the ester linkage part. Okay, so you guys know that there's different kinds of fats, right? Like it could be butter. That was kind of the reason that you did that exercise. So like butter is a fat, lard, you can buy lard in a bucket, that's a fat. Crisco, that's a fat. We have like olive oil, that's a fat. Margarine, there's all kinds of things that are fats, peanut oil, there's all kinds of different fats. So remember, all these have in common that they're water insoluble because they have that nonpolar carbon and hydrogen. Carbon is 2.5, hydrogen is 2.1. Difference between these is 0.4, which is less than 0.5, and that makes it non-polar. And if it's non-polar, remember, water can only dissolve things that have a charge. This has no charge, therefore it's not dissolvable in water. And we use the term hydrophobic, water-fearing, to describe things that are not dissolvable in water. So you probably heard this term. You probably heard the term saturated versus unsaturated fat. And in fact, I would bet if you went and looked at your nutritional label on a bag of peanuts or something like that, you would see that there's some saturated and unsaturated fats. And you probably know that the saturated fats, everybody says they're bad for you, and the unsaturated fats are fine. So why is that? Well, we can talk about it, but let's talk about what saturated means. So saturated means that it has the maximum number of hydrogens that it is allowed to have. So this, if we had 2 carbons, we know carbon can make 4 bonds. Each carbon has 4 bonds it can make. So we have, this carbon has as many bonds as it can form. And besides this bond to link it to another carbon, all the other ones are attached to hydrogens. Same thing here. So this is what we call a saturated molecule. We could not add another hydrogen to this molecule and still follow the rules of chemistry. This one has a double bond in it. And that double bond, because carbon can only form 4 bonds, we've got 1, 2, 3, 4. That means it causes it to have less hydrogens than the maximum, because if this was a single bond, it would have 6. But this one only has 4 because of that double bond. Because it doesn't have as many hydrogens as it can have, then it's considered unsaturated. whereas this would be called saturated. Do you guys get the difference between the two? So if you just want to simplify it, you could say throwing a double bond in there makes it unsaturated, whereas not having any double bonds makes it saturated. All right, so what's the big deal and the difference between these two? So saturated fats are usually solid at room temperature. So think of like butter, margarine, lard, things like that. Those are saturated fats. Usually these come from animals. Remember, there's no double bonds. Otherwise, if there was a double bond, it would be called unsaturated, right? So it has the maximum saturated number of hydrogens. And if we were to draw this out, we could draw, I don't want to draw a gigantic chain, but it would have the maximum number of hydrogens we can fit on here. Sorry, it sounds like I'm running a daycare, but I'm not. My brother-in-law and his wife are here with their baby visiting, so there's a bunch of babies here. Anyway, so this is what a saturated would look like, but what's important is that it makes this straight chain. Remember we talked about the shape is super important. This is that glycerol head. So this is the glycerol, and that's where it would attach, and this is a straight chain. So remember, you have 3 fatty acids, so you have 3 straight chains with saturated fats. If we look at unsaturated fats, they have 1, and we call that mono, or more, so we call that poly. So you probably heard mono unsaturated fat on a molecular level. That means it has a single double bond, or a poly unsaturated fat on a molecular level. That means it has more than 1 double bond, so 2 or more. They're liquid at room temperature. They're mostly from plants, and these double bonds cause it to bend. So on a molecular level, it would look sort of like this, where you would have, remember we talked about that this can't rotate? And so before I drew it out like this, and we just had, I just had a simple hydrogen on there. Dang it. Remember, and I asked you, is this cis or trans?
Cis.
Yeah, it's cis, because the long chains, the unusual functional groups are on the same side. And so it's the same thing if I add, instead of having these hydrogens on here, if I added a long chain of carbons on it. So the fatty acid would look something like this. It would have, I'm not going to draw all the carbons, but let's just say that it's a 6 carbon. It would be much longer than that, but I don't want to draw the whole thing out. And so this carbon has 3 bonds on it. So it would have a hydrogen here, and this one would also have a hydrogen here. Does that make sense? And so if you look at the shape of it, you can see that it's bent like this. Does that make sense to everyone?
Yeah.
But this is a cis fat, right? Because these long chains are on the same side, and these hydrogens are on the same side. So this is a cis fat, but it's also unsaturated because it's bent. Now here's the difference. You have these straight chains, right? And then you have these bent chains like so. And then you have your glycerol head. So which one of these do you think takes up more space? If I were to shrink myself down and look at them, which of these molecules do you think are bigger?
The bent.
The one with the bent chains, right? So think about it like this. Let's say, you're out playing in the snow and you want to make a snowball. Are you just gonna pick up a handful of snow and throw it at your friend?
No.
What would you do?
Pack it.
Right. You're packing it, and what you're doing is you're compressing it. So once you compress it, does it get more solid or less solid?
More solid.
Yes. And so these, because they're straight chains, we can compress them like stacks of paper on each other. Because these are bent, it would be like crinkling up paper and stacking it together. They're not as dense, right? They're not as compact. And that's why this is solid, right? These saturated fatty acids are solid at room temperature, where these are liquid, because saturated are straight chains and they're more compact. And these are less compact. Does that make sense?
Yeah.
Okay. So if you're a manufacturer of stuff like ice cream and things like that, you want your ice cream to be relatively solid. You don't want it to be liquidy. And so in order to do that, you would need to get animal fats. What do you think is easier to get more of? Plant fats or animal fats?
Animal fat.
Well, think about how many animals there are that you can get fats out of and how many plants there are you can get fats out of.
Oh yeah, true. So plants.
So there's going to be a lot more plants. So it's much easier to grow a field of corn than to grow a field of cows. And so people that make things that they want to be more solid at room temperature, like butter and stuff like that, they want to take the liquid oil of unsaturated fats and turn them more solid. So there's ways to do that. So if we looked at it, we know that this is going to be saturated. It's a straight chain, right? And it's going to be solid. But I have this, which is going to be bendy, and this is going to be unsaturated. So how do I turn my corn oil into something more like butter?
Can you remove the double bond?
Yeah. And if I do the remove the double bond, what do I have to add onto this molecule?
More hydrogen?
Yeah. So you ever heard of the term hydrogenated?
Yeah.
So when a fat is hydrogenated, it's basically turning a liquid fat into a solid fat. So they can put that in your ice cream, or they can turn vegetable oil into margarine, right? Because that's made out of vegetable oil, whereas butter would be made out of animal fat. And that's why a lot of people are saying, well, butter is better for you because it's more natural. This is artificial. They're taking liquid and making it solid. So the other thing is that you might want to, let me erase this. There's another trick you could do instead of adding hydrogens to it. So remember, the goal is the shape, right? We want it to be flat so we can compact it together. So let's go back to our bent shape, which is the cis fat, right? And instead of making it cis, so it would be, this would be on the same side, we make it like this. So what is this shape more like?
It's a little more straight.
Right, it's more straight, it's not bent. And if it's more straight, when we add it onto that glycerol, we can pack it tighter. And so this is a trans fat. So what do you think is going to be more solid at room temperature? Cis fats or trans fats?
Trans fat.
Yeah. So manufacturers take cis fats and they turn them into trans fats, so they're more solid. So your ice cream is more creamy and your margarine is more margarine. Does that make sense?
Yeah.
All right. So what I want you guys to get out of this is you should know the difference between saturated and unsaturated fats, right? It's pretty simple. Saturated, just think about, has the most hydrogens that you can possibly put on it. It can't have any double bonds. Unsaturated has to have at least one double bond. If it has one, it's monounsaturated. If it has two, it's polyunsaturated, or it could have more. It could have three, it's still poly. That saturated fats are solid at room temperature and they come from animals. Unsaturated fats are liquid at room temperature and they come from plants. The cis and trans thing, I don't care that you know that. I'm just showing you why trans fats are bad. What would you rather have flowing through your blood? Something that's solid at room temperature or liquid at room temperature?
Liquid.
Yeah. So that's why trans fats and saturated fats are generally bad for you because they can clog up your arteries. All right. But fats have purpose. We need fat to store energy. If you don't have enough fat, you can't synthesize things like steroids, which can be really bad. So people that these bodybuilders that go on these extreme diets and have super low body fat, they can't produce testosterone anymore. Female bodybuilders can't produce estrogen. They can't produce any hormones because they don't have any fats to do it. And so, this can cause like weakness, like, they may look big and strong and tough, but they're actually at their weakest whenever they look that way. This can cause like problems with your menstrual cycle and stuff like that. In fact, many female bodybuilders, because they don't produce enough estrogen, will actually stop their menstrual cycle until they restore like a 10% more healthy body fat as opposed to like a 4 to 5% body fat range. We talked about the reason animals use fat because it has about twice, not quite, 4 to 7 calories per gram than carbs. And we talked about that this is beneficial for animals so that they weigh less, so they can run faster when the cheetah's, after you or whatever. As opposed to plants who really don't care what they weigh because they're not going anywhere. Unless you guys have seen plants running around lately. And then I would suspect if you found plants that moved around on different planets, they probably would use fat as an energy source instead of carbs. Fats are important to cushion vital organs. You guys ever watch like MMA fights and stuff like that? Your kidneys have a big layer of fat around them. If they didn't, those guys are punching each other in the kidneys. They would be peeing blood for a month. So you need some fat to cushion your organs. Remember, your body cavity is hollow. And then, certain organisms need it to prevent heat loss, like things that live in the Arctic or in cold climates. Whales, because they're really deep in the ocean. Sea lions, walruses, polar bears have a lot more fat than their grizzly cousins. So fat's important for allowing animals to live in colder climates. Any questions about that? All right. So the next thing that we're going to talk about are the phospholipids. So phospholipids are just like triglycerides, except on this carbon. So normally you would have 3 fatty acid tails, but in the phospholipid, instead of having that third fatty acid tail, it has what we call a phosphatidylcholine head. I know that's a big name, and I don't care that you know that. What I do want you to know is, remember this phosphate has a charge, and we pointed this out in chapter 4. So if it has a charge, is it water soluble or not water soluble?
Water soluble.
Right. So this part of the molecule is water soluble, which we call hydrophilic. So it's hydrophilic, right? And this part of the molecule is made out of fatty acid tails. So is this water soluble or not? Nonpolar.
No.
So this is one of those amphipathic molecules that we talked about. It has a dual nature. One end is water fearing and one end is water loving. And this is really important in the formation of cell membranes. So phospholipids are found in cell membranes. And we talked about this. We don't want to build our house out of paper, right? Because if it rains, it would melt. We don't want to make our cell membranes out of something that is water soluble, because when it's around water, it would dissolve our membrane. And then we couldn't maintain, we'll take a little journey back to chapter 1. What is it called maintaining a relatively constant internal environment?
Homeostasis.
Right. So if we didn't have the cell membrane, we couldn't do homeostasis. And we define this as a requirement for living things way back in chapter 1. So without a cell membrane, life wouldn't exist. And without phospholipids, you couldn't have a cell membrane. And this is why. So phospholipids, and this is just an artist drawing. So this is the water-loving head, and these are the fatty acid tail. So when you put these things in water, they always line up this way. So the heads, they're hydrophilic. So they're always going to line up towards water. And the tails are hydrophobic. So they're always going to line up away from water. So this is an automatic thing. If you put a phospholipid in a glass of water and you shook it up, it would form this structure. This structure is called a micelle. You should probably know that. That's a term that's commonly used in biology. But this is the structure. And sorry, those are my dogs. I must have a delivery or something. Anyway, so this is inside here, there's no water. And you guys know that water is required for life. So this structure actually wouldn't be conducive to living things, because there's no way to get water on the inside of this sort of, cell. It's not a cell, it's called a micelle. But the way to do this is we use 2 layers. So we can think about it this way. This is linear, but imagine this curves around and makes a cell. So we're just looking at part of it, like part of your cell membrane. And so here we have the fatty acid tails going in and the hydrophilic heads going out. So this is water out here and water in here. And because of this structure, because it's what we call a bilayer, so that means 2 layers, we can have water on the inside and water on the outside. And in between, it's fat. So water can't cross that. So because of this dual layer, we can have a cell membrane that prevents anything from the outside coming on the inside. So we can maintain homeostasis. And all living things, no matter what it is, bacteria, a frog, a plant, they all have cell membranes that are composed exactly out of this. And remember we said the smallest thing that has all those properties of life is a cell. That's the cell theory. So all cells are made up of what we call phospholipid bilayers. So a phospholipid bilayer is 2 layers of phospholipids that allow to have this fat layer in the middle that water can't cross, and that allows for homeostasis, but it allows for a water environment on the inside of the cell and a water environment on the outside of the cell. And this allows us to move things around inside the cell more easily than if it was air. In fact, I think I used this analogy before, but let's say that I had a bag of sugar in the corner of a room chained to the floor, and you chained to the other end. You wouldn't be able to get to that sugar, but if I filled that room with water, like up to your neck, the sugar would dissolve, and then eventually it would transfer over to you, and you could drink it, and that would allow you to get energy. Whereas there's no way that that sugar would fly through the air and come to you. But water can move things around, and that's why we need water to live. Okay, any questions about those? So we talked about the triglycerides. And what are those made out of? The monomers are glycerol. Most of you guys got this right on that table in the lab. And fatty acids, how many?
3 fatty acids.
Awesome. And so these fatty acid chains can be saturated, and what does that mean? Are they solid or liquid at room temperature?
Solid.
Perfect. And if they're unsaturated, then they're liquid. What causes it to be unsaturated?
They have a double bond.
Perfect. And then we can have trans or cis, which you don't need to know except for chapter 4, but that changes its shape to be linear or bent, and that's why it's liquid or solid room temperature. I'm going to go back for a second. I think I skipped a slide that I meant to go back to. Maybe I'll come back. Maybe I'm going to come back to this. Anyway, so the next group, so we have triglycerides, we have the fatty acids, and we have the phospholipids. And because this phosphate has that negative charge, has a water-soluble head, and it has fatty acid tails. How many fatty acid tails does this have? This has 2. Remember that third one is replaced with that glycerol phosphate group. And then the third group we have is the steroids. And the steroids are lipids with a carbon skeleton that have 4 carbon rings. And you can see this here. So this is cholesterol. I know you guys have heard that cholesterol is terrible for you and all that stuff. But what you're really talking about is HDL and LDL. And LDL stands for low density lipoprotein. So the name tells you what it is. This isn't a fat. The cholesterol that they talk about on TV and stuff that's bad for you, that you hear from your doctor, that's really a protein, and its job is to carry fats because they're not water soluble. So this thing is a fat carrier, protein, not really cholesterol. Cholesterol isn't water soluble. It's not a protein. It doesn't have amino acids in it. And cholesterol is very important because it is a precursor for all your steroid hormones. So testosterone, estrogen, cortisol, and you guys will learn all about these in anatomy and physiology, but there's literally hundreds and hundreds of different steroids. They're all made from cholesterol, and they all have this basic 4-ring structure. In fact, I'm going to show you. So when I worked at the VA, part of my job was to do research on why everyone said that cholesterol causes cardiovascular disease. So my job was to look at various of these cholesterol steroids to find out if they cause cardiovascular disease. So you probably heard on the news that LDL is bad for you. There's a thing called the Framingham study. And what it did is it followed veterans for, since the First World War and looked at them as a population. So some people have normally high LDL and some people have normally low LDL. And so you probably heard that LDL causes heart disease. But if that's true, if you followed people that had normally high LDL and people that had normally low LDL, it didn't matter what they ate. Only 25% of people can change their LDL by diet. What would you think? The people with high LDL, would they have more or less heart attacks?
High LDL had more heart attacks.
And low LDL should have fewer heart attacks. But if you look at the Framingham study, guess what? They're the same. So what does that tell you?
It has nothing to do with heart health.
Right. And so this myth has been perpetuated by the pharmaceutical industry and by doctors and cardiologists for years. And I don't understand it. The closest I can come to is that there was a study on rats years ago that showed that LDL made a difference, but in humans, there's really absolutely no evidence. You guys can look this up for yourself if you don't believe me. It has to do with the ratio of LDL to HDL, and it turns out that HDL actually has what we call an antioxidant property, and it's really the antioxidants that can affect your cardiovascular, the rate of your cardiovascular events. In fact, so I'm just going to look this up real quick, and then I'll end the lecture and jump over to office hours. So in the beginning, when Pfizer made Lipitor, which lowers your LDL, they had to put a disclaimer at the bottom that said there's no correlation between cardiovascular disease and LDL levels, what they call cholesterol levels, bad cholesterol. I actually was part of a study that was funded by Pfizer that showed that taking Lipitor reduced cardiovascular events, but it's kind of shady the way that they worded this, and the FDA allowed them to take that disclaimer off. But it's kind of like saying eating ice cream in the summer increases in Phoenix and so did drowning. So ice cream must cause you to drown. What happens is that steroid reduces your LDL, which increases your HDL to LDL levels. And that HDL then protects you from cardiovascular events. So it's not the LDL that's doing it, it's the antioxidant protective effects of the now increased ratio of HDL. Does that make sense? That causes you to not, so it's not the cholesterol, it's the antioxidant property of Lipitor that prevents you from having cardiovascular disease, not the part that lowers the cholesterol. Anyway, so we can just search for if we look at all these different kind of androus or cholesterol or estero or whatever based or pregnant, you probably heard of like Pregnisol or whatever. So all of these steroids, you can see, and the reason I want to show you is that all of these have this basic 4-ring structure. You guys see my screen?
Yeah.
And so all of these steroids come from cholesterol. And so you can see that these are the functional groups that we can add to them. And then this changes all of their properties and stuff like that. And the reason I'm showing you this is because I don't want you to memorize the hundreds of thousands of different steroids. But what I want you to see is that they all of these steroids have that basic 4-ring structure that comes from cholesterol. So the moral of the story is, without cholesterol, you wouldn't be able to make any of your hormones, your steroid hormones. Any questions about any of that? I'll take that as a no. All right, so I'm gonna stop lecture now, and I'm gonna jump over to office hours. So I might see you there. Otherwise, I'll see you tomorrow at office hours or Thursday for lecture. Okay.
Thank you, Professor.
All right. Have a good one. Bye.
You.